as many as 100 million different living organisms, but fewer than 10% have been discovered, much less identified and classified. If we organize these microbes based on their similarity or differences in any characteristics, we can easily contemplate various microorganisms. The objective of taxonomy is to classify living organisms, that is, to establish the relationships between one group of organisms and another, and to differentiate them. In this chapter, we will learn the various classification systems, the different criteria used for classification, and the tests that are used to identify microorganisms that have already been classified. To facilitate research, scholarship, and communication, we use taxonomy to show degrees of similarities among organisms. That is, we put organisms into categories or taxa. The formal system for organizing, classifying, and naming living things is taxonomy. This science originated more than 250 years ago when Carl van Linne laid down the basic rules for taxonomical categories or taxa. The primary concerns of taxonomy are classification, nomenclature, and identification. These three areas are interpreted and play a vital role in keeping a dynamic inventory of the extensive array of living things. Classification is the orderly arrangement of organisms into groups that indicate evolutionary relationships and history. Nomenclature is a process of assigning names to the various taxonomical ranking of each microbial species. Identification is the process of determining and recording the traits of organisms to enable them placement in an overall taxonomic scheme. The main taxa or groups in a classification scheme are organized into several descending ranks called hierarchy. It begins with domain, which is a giant all-inclusive category based on a unique cell type, and ends with species, the smallest and most specific taxon. All the members of the domain share only one or few general characteristics, whereas members of a species are essentially the same kind of organisms. That is, they share the majority of their characteristics. The order of taxa between the top and bottom levels is in descending order: domain, kingdom, phylum or division, class, order, family, genus, and species. Thus, each domain can be subdivided into a series of kingdoms. Each kingdom is made up of several phyla. Each phylum contains several classes, and so on. Because taxonomic schemes are to some extent artificial, certain groups of organisms don't exactly fit into the 8 taxa. In that case, additional levels can be imposed immediately above or below a taxon, giving us such categories as superphyla and subclass. The way we have classified organisms has changed greatly over the centuries. Comparing the sequences of nucleotides in ribosomal RNA from different kinds of cells shows that there are 3 distinctly different cell groups: the eukaryotes and 2 different types of prokaryotes, the bacteria and the archaea. In 1978, Carl Woese proposed elevating the three cell types to a level above kingdom, called domain. The evolutionary relationship of the three domains is the subject of current research by biologists. Based on RNA analysis, three-cell lineage clearly emerged as cells were forming 3.5 billion years ago. That led to the archaea, the bacteria, and what eventually became the nucleoplasm of the eukaryotes. However, the three cell lines were not isolated from each other. Horizontal gene transfer appears to have occurred among them. Analysis of complete genomes shows that each domain shares genes with other domains. In addition to differences in RNA, the three domains differ in membrane lipid structure, transfer RNA molecules, and sensitivity to antibiotics. Microbial taxa within bacteria and archaea form discrete genealogical cluster groups that can be illustrated in phylogenetic trees show inferred evolutionary relationships in the form of multiple branching lineages connected by nodes. The organisms whose nucleotide sequences have been analyzed is identified at the tip of each branch. Each node represents a divergence event, and the length of the branches represents the number of molecular changes that have taken place between the two nodes. A tree may be unrooted or rooted. An unrooted tree simply represents phylogenetic relationships but doesn't provide an evolution path. Figure A shows that A is more closely related to C than it is to either B or D, but it doesn't specify the common ancestor for the four species or the direction of change. In contrast, the rooted tree gives the node that serves as the common ancestor and shows that development of the four species from this root. It is much more difficult to develop a rooted tree. For example, there are 15 possible rooted trees that connected four species but only 3 possible unrooted trees. Living organisms are grouped according to similar characteristics, and each organism is assigned a unique scientific name. The system for classifying and naming, which are used by biologists worldwide, is called binomial nomenclature. Every organism is assigned 2 names, or binomial. These names are the genus name and the specific epithet, and both names are printed, underlined, or italicized. The genus name is always capitalized and is always a noun. The species name is lowercase and is usually an adjective. This table contains more examples. Binomials are used by scientists worldwide, regardless of their native language. This nomenclature enables them to share knowledge efficiently and accurately. That's all. Thank you for your attention.